Welcome back. Let's continue exploring the fascinating world of cell organelles and uncover more about the structures that keep the cell functioning. In this part, we dive into ribosomes, the molecular factories that drive protein synthesis. Ribosomes are tiny dense granules composed of ribosomal RNA and proteins, serving as the site of protein synthesis by translating messenger RNA into polypeptides. Ribosomes are roughly spherical, granular, non-membranous bodies found in both eukaryotic and prokaryotic cells. Ribosomes were discovered by George Emile Palade in 1955. Each eukaryotic ribosome consists of two subunits. The larger subunit sediments are at 60 Svedberg units, while the smaller subunit sediments are at 40 Svedberg units. Two subunits after attachment form an ADS particle. The attachment is controlled by the presence of magnesium ions concentration or forming salt bonds between the phosphate group of RNA and an amino group of amino acid or both by magnesium ions and salt. However, eukaryotic ribosomes are larger and characterized as ADS ribosomes, while the prokaryotic ribosomes are slightly smaller and are characterized as 70S ribosomes. Eukaryotic ribosomes are composed of almost equal RNA and protein. Hence, they are ribonucleoprotein particles. The RNA present in ribosomes is called ribosomal RNA. Do you know where ribosomes are formed? Ribosomes are formed in the nucleolus. Then these are transported to the cytoplasm through the nuclear pore. Ribosomes exist in two forms, either freely dispersed in cytoplasm or attached to rough endoplasmic reticulum as tiny granules. The nucleolus is the factory of ribosomes, while ribosomes are the site of protein synthesis. Golgi complex or dictyosome. The Golgi complex is a membrane-bound organelle that modifies, packages, and transports proteins and lipids, received from the endoplasmic reticulum for secretion or use within the cell. The Golgi complex is present in all eukaryotic cells and was discovered by Italian biologist Camillo Golgi in 1898. The Golgi complex comprises a stack of flattened, membrane-bound sacs known as cisternae. These cisternae, along with associated vesicles, form the Golgi complex, a system of interconnected tubules surrounding the central stack. At one end of the stack, new cisternae are constantly formed by the fusion of vesicles from the smooth endoplasmic reticulum. The Golgi complex has two distinct faces, the outer forming face known as the cis face, which is convex, and the inner maturing face, or trans face, which is concave. At the trans face, the cisternae break up into vesicles. The cis face of the Golgi, near the endoplasmic reticulum, receives proteins, starts their modification, and sorts them, while the trans face, near the plasma membrane, finishes processing, packages proteins into vesicles, and sends them to their final destinations. The main function of the Golgi complex is to process cell secretions, which is why it is abundant in secretory cells. It collects proteins from the rough endoplasmic reticulum via the smooth endoplasmic reticulum, modifies them to perform specific functions, and then exports these modified products in the form of vesicles. An example of this is in mammals, where the pancreas secretes granules filled with digestive enzymes to assist in digestion. Certain organelles, such as lysosomes, peroxisomes, and glyoxysomes, also originate from the Golgi complex. Golgi complex is also involved in the formation of conjugated molecules like glycoprotein, lipoprotein, etc. In plant cells, during cell division, the Golgi complex generates vesicles containing materials for cell wall synthesis. During cytokinesis, Golgi vesicles arrange at the cell equator and fuse to form a structure called the phragmoplast, from which the new cell wall is derived. The phragmoplast acts as a scaffold for the assembly of the new cell wall during cytokinesis. Lysosomes are membrane-bound organelles found in most eukaryotic cells, containing digestive enzymes that break down phagocytosed food particles, cellular waste, and foreign substances. Any foreign object that gains entry into the cell is immediately engulfed by the lysosome and is completely broken into simple digestible pieces. This process is known as phagocytosis. Lysosomes are most abundant in animal cells that exhibit phagocytic activity. Lysosomes are single membrane-bound sacs rich in acid phosphatase and various hydrolytic enzymes. These enzymes are synthesized in the rough endoplasmic reticulum, processed in the Golgi apparatus, and then bud off as primary lysosomes. Lysosomes also release enzymes for extracellular digestion. Ingested food is enclosed in food vacuoles, which fuse with lysosomes to form secondary vesicles, where digestion begins with the help of enzymes. The digested nutrients are absorbed into the cytoplasm, while the undigested waste is enclosed in contractile vacuoles, which fuse with the cell membrane and are excreted through exocytosis. Do you know why lysosomes are called suicide sacs? Under conditions like starvation or normal cellular processes, primary lysosomes engulf and digest parts of the cell to generate energy. These specialized lysosomes, called autophagosomes, along with digestive vacuoles, are collectively known as secondary lysosomes. Lysosomes help recycle cellular materials by digesting old and worn-out organelles like mitochondria, contributing to cell renewal. However, their enzymes can also lead to cell degeneration, which occurs in some developmental processes. Autolysis: during development, cells undergo programmed cell death, where lysosomes rupture, releasing digestive enzymes into the cytoplasm. This leads to cell breakdown into smaller fragments, which are then engulfed and cleared by other cells. Lysosomes are known as suicidal bags of the cell due to their self-destructive potential. Lysosomal storage diseases. Storage diseases occur due to the accumulation of substances like glycogen or glycolipids within cells, caused by mutations in lysosomal enzymes that impair the breakdown of these substances. One example of a storage disease is glycogenosis type 2. In this condition, the liver and muscles become overloaded with glycogen trapped in membrane-bound organelles. This happens because the enzyme responsible for breaking glycogen into glucose is missing. So far, around 20 similar diseases have been identified, each caused by the absence of a particular enzyme. Tay-Sachs disease occurs due to the absence of an enzyme required for lipid breakdown. The accumulation of lipids in brain cells leads to mental retardation and can result in death. Krabbe disease occurs when the body can't make an enzyme called galactosylceramidase, leading to the loss of myelin, the protective coating on nerves. This results in nerve damage, preventing the brain from sending signals to the body and causing Krabbe disease symptoms. Gaucher disease is a rare inherited disorder where a missing enzyme prevents the breakdown of lipids, causing them to accumulate in organs like the spleen and liver. A vacuole is a membrane-bound organelle involved in storage, waste disposal, and maintaining turgor pressure. It stores water, nutrients, and waste products. Vacuoles exist in both animal and plant cells. In mature plants, a large central vacuole is formed by the fusion of smaller vacuoles containing cell sap that serves as the primary reservoir for inorganic ions like potassium and chloride. The vacuole is enclosed by a membrane called the tonoplast, which separates it from the cytoplasm. Let's take a quick look at the important roles vacuoles play in cells. Food vacuoles. The food vacuole stores and digests nutrients within the cell. Maintaining water balance in freshwater. Protists. In freshwater protists, contractile vacuoles help remove excess water, maintaining the proper concentration of ions and molecules inside the cell. Protecting plants from herbivory. The storage of harmful and dangerous compounds in vacuoles protects plants from herbivory by deterring herbivores. Maintaining turgor pressure. The large central vacuole plays a crucial role in maintaining turgor pressure and acts as a storage site for the cell. Mitochondria are essential organelles with a double membrane, responsible for ATP production through oxidative phosphorylation. This is why they are often called the powerhouse of the cell. Cells that require more energy, such as muscle and nerve cells, contain a higher number of mitochondria to meet their ATP needs. Mitochondria have their own circular DNA and can replicate independently through binary fission, just like prokaryotic cells. Mitochondria are cylindrical, rod-shaped organelles enclosed by a double membrane. The outer membrane of mitochondria is smooth, while the inner membrane forms folds called cristae. The space between these membranes is known as the intramembranous space. The outer mitochondrial membrane is smooth and porous, permitting molecular exchange between the cytoplasm and the intramembranous space. The inner mitochondrial membrane is selectively permeable and folded into cristae, which increase the surface area for efficient ATP production through oxidative phosphorylation. The mitochondrial matrix, enclosed by the inner membrane, contains ribosomes, mitochondrial DNA, and RNA, supporting protein synthesis and metabolic processes. Do you know? Like a legacy passed down through the mother's line, mitochondria are inherited only from the mother, never the father. Next, we'll look at the roles mitochondria play in energy production and cellular processes. Krebs cycle. The Krebs cycle, occurring in the mitochondrial matrix, processes acetyl-CoA to generate energy-rich molecules like NADH and FADH2, which fuel ATP production in the cell. Aerobic respiration. Mitochondria are the site of aerobic respiration, where oxygen is used to efficiently convert glucose into ATP, producing carbon dioxide and water as byproducts. Fatty acid metabolism. Within mitochondria, fatty acids are broken down via beta oxidation into acetyl-CoA, which enters the Krebs cycle, providing a crucial energy source for the cell. Electron transport chain. Located on the inner mitochondrial membrane, the electron transport chain transfers electrons from NADH and FADH2, creating a proton gradient that drives ATP synthesis via oxidative phosphorylation. That wraps up the second part of our discussion on cell organelles. In the next segment, we'll cover the remaining organelles and their vital functions. If you found this helpful, don't forget to like, share, and subscribe for more videos.